Thank you. I hope everybody will be have taken the leave and rest period is finished. And now we have started to listen from the gut health from the professor. Thank you very much for your wonderful talk. And now I will continue with the important issue that is not only a Pakistan perspective, but this is a global issue of mycotoxins. Actually, the story of mycotoxins start from the fungal contamination of the grains, especially, and this fungal contamination downgrade the commodities. And ultimately, there is a many times there is a overlap of the food chain. Many times, some staple cereals are shared between the human and animal and the feed industry. So every nation has a priority to select the good quality grains for their human population. So sometimes compromised grains are diverted toward the poultry industry. And it will lead to the production of mycotoxins, and the toxigenic fungi produce a variety of mycotoxins. And all toxigenic fungi are not producing mycotoxins. Generally, the main fungal contamination species, especially with reference to the our region, Aspergillus, Penicillium, Fusarium, and Alternaria. You can say among these Aspergillus, Penicillium, Alternaria, these are mainly storage fungi. And this storage fungi that will affect if your storage quality is poor. So this is a very important issue. Then these fungi infect the cereals, and ultimately it will lead to the production of mycotoxins. There is a condition favorable for the fungal growth if relative humidity is high over 70% for few days to few weeks, and temperature more than 26 degrees Celsius, and high moisture content of the crop. This is very important that we are facing. If there is a high moisture content, many times these grains also bring low price. Nowadays, our bigger feed mills, they have a dryer, but still all the feed mill systems don't have this facility. So high water content is very important. This is one of the factors that favor the fungal contamination and ultimately production of mycotoxins. And other factors, stress factors also include drought, flood, or insect infestation. These also that you can say these are the predisposing factors. These mycotoxins are the secondary metabolites produced by these toxigenic fungi, and these are carcinogenic, teratogenic, mutagenic, and non-immunosuppressive agent nowadays. And these are cytotoxic and very important. Mycotoxins are heat stable. Even your roasting temperature doesn't destroy mycotoxin. It can decrease the level, but it will not eliminate the mycotoxins. And significant economic losses affecting the poultry and human health, because this is one of the important health issue also. And poultry performance is badly affected if you have high level of mycotoxins, and impact on the national as well as international trade. Because I was discussing with the colleagues that some people from Pakistan, they exported the milk to the Malaysia, and those containers were rejected due to high level of aflatoxin in the milk. That was aflatoxin M1. That is a metabolite of B1. So this is very important, because now importing countries, they have certainly regulatory limits, and then they check your product for the mycotoxin. So it is directly affecting your international trade. Among these mycotoxins, aflatoxin is one of the most important and most toxic compound. And there are 4 types of aflatoxin: B1, B2, G1, and G2, named on the basis of their fluorescence under the UV light. And aflatoxin M1, actually this is a B1, but this is a metabolite, and it is named with reference to the milk as M1. And major fungi that produce aflatoxin include Aspergillus flavus, produce B1 and B2, and Aspergillus parasiticus produce all 4, B1, B2, G1, and G2. And aflatoxin M1, this is a class 1 human carcinogen according to the International Agency on Research on Cancer. So there is a study. If your food is contaminated with biological vectors, hepatitis B and C, and if same time you have a high level of aflatoxin, the chances of hepatocellular carcinoma increase to 60 times. So this is a very important. This is a one of the known 10 factors mentioned by the WHO for the liver cancer in human, and aflatoxin B1 is one of them. Then ochratoxin, this is famous for the Balkan nephropathy. That was a huge loss in the human population in Balkan countries. Ochratoxin A, B, and C, and among these, ochratoxin A is most toxic one. And this is the major toxin producing fungi include Aspergillus ocraceus, Aspergillus carbonarius, and Aspergillus niger. These are the black Aspergilli. Many times you see in even our buildings there is a contamination of black color spores. And ochratoxin A is a possible class 2B human carcinogen according to IARC. And among these toxins, the aflatoxin B1 targets the liver, but it doesn't mean aflatoxin B1 will spare the kidneys. Although its target organ is liver, and it leads to the hepatotoxic and carcinogenic. And ochratoxin A is a nephrotoxic and carcinogenic, and this is known for kidney failure. And fumonigens are teratogenic and carcinogenic. Then don toxin is deoxynivalenol. This is it lead to gastroenteritis, and zearalenone is very important in dairy animal. It is leading to the hyperestrogenism. And T2 toxin induce gastric ulcers and enteritis, and pigs are also very sensitive to the T2 toxin. Mycotoxin in the feed. This is the second most issue faced by the global poultry industry after the high feed prices. If we give numbering to the issues, first important issue of the industry is high feed prices, and second important issue is the mycotoxin, because these are the unavoidable contaminant of the agriculture commodities. And mycotoxins, contaminated feed are big risk to the animal health. Contaminated feed may result in the residues in the food obtained from the animal origin like milk, meat, and eggs. And ultimately, it will affect the end users. So this one slide represents how these mycotoxins are a one health issue. If there is a contamination or infection of the plant or crop, ultimately these will go to the one fate is that these will go into the animal feed, and contaminated feed will ultimately consumed by the animals or birds, and it will go into the toxins in the blood and organs, and these animal origin products are consumed by the human population. And the second way is that these grains are directly consumed by the human population, although this is less in Pakistan, but in Asia Pacific country, they mostly use boiled corn with cheese. So many times, I mentioned in the earlier slide, even roasting temperature doesn't destroy mycotoxins. And so, and third way is that through the milk of the dairy animal, it is ultimately consumed by the end user. So these are the possible way, and this is a classical example: environment, animal, and human. So this is a classical triangle of the One Health. This is a briefly how these mycotoxins lead to the toxicity. This is a general mechanism of toxicity that is almost good for the all mycotoxins. That from the environment ultimately it lead to the reactive intermediates. But the body has a mechanism. Nature has a mechanism that metabolic detoxification and excretion. And if toxin load is high, it lead to DNA death. That are changes the normal physiology of the DNA, but still body has a system of DNA repair. That is a natural mechanism. But if damage is still more, then ultimately there are two fate: genetic damage. And genetic damage will lead to the cell death or purposeless proliferation of the cell that we name as neoplasia or cancer. This is our recent publication that was on the risk assessment of the hepatocellular carcinoma with aflatoxin B1 exposure in the edible oils. This is a very interesting study that the edible oil we are consuming in Pakistan, those are from our meals, mostly our oilseed crops. If they are having high level of aflatoxin, ultimately there is a risk to the human population, because Pakistan one of the leading oil consuming country, and mostly we are importing the oil. So this is a recent survey by DSM And if you will see these, I have highlighted these red. That is for the South Asian region, that aflatoxin contamination up to 70%, zearalenone 30%, T2 64%, formonicin 60, and ochratoxin 66%. This was a huge study. They collected around 11,000 samples from the different countries, DSM. So, and there was the issue of multiple mycotoxin. This is another important issue, because if there is a multiple mycotoxin issue, they will potentiate the toxic effects. So if there is a fungal contamination, that is a very basic thing. Many times we observe on the corn, you can see this fungal contamination, fungal growth, and very basic criteria. Like nowadays, we check the currency notes, we put these grain under the UV light. If there is a fluorescence, that is a basic criteria, this is having a toxigenic fungi. If these grains will go into the feed, ultimately this type of the feed there will be, and fungal growth is always in pocket. Very interesting and tricky thing in the mycotoxin is the sampling. Many times we do sampling in the feed mill, we take sample randomly from one bag. In a one feed bag, there is a huge variation. One bird may have a 5,000 ppb level and other bird will remain okay. Because always you have to thorough mixing, then you have to collect the representative sample. Many times fungal growth is in the form of pockets. And if such feed will be consumed by the bird, so ultimately you can see this type of lesions will be developed in the bird. And liver is the chief metabolizing organ of the body, chief gland of the body. And if liver health will be compromised, then you can assess bird health will be compromised. So the upper one, you can see typical lesion for aflatoxin. There is anemic appearance of the liver. Definitely, we all observe lower body weight, lower feed intake, and egg production will drop. And microscopically, there will be in the grossly in the kidneys, there will be pale, large kidneys. These are lesions many times we see on the necropsy on the form. And if we go for the histopathology, you can say upper one, classical fatty change or liver. So you can see almost all the parenchyma is having vascular degeneration, and lower one is the normal picture of the liver. You can compare both one. Similarly, in the kidneys, those will be swollen, bulging out from the bony socket. I think such kind of kidneys are self-sufficient to kill the bird. There is no need of any other ligand. And in the lower one, you can see the histopathology. There is a severe congestion and nephrotic changes, severe nephritis that will be developed by the mycotoxins in the kidneys. This is a very interesting, that paper we published in the toxicologic pathology in the US journal. You can see in the left one, this is the normal neural tube of the chicken. This is the impression of vertebrae. This is the normal neural tube. This is a normal and proper closure. And the right one, you can see improper closure. Improper closure will lead to the skeletal abnormalities in the bird. And these are the teratogenic effect. If high level in the feed of breeding hen, it will lead to skeletal abnormalities in the chicks. So this is the teratogenic effect of the mycotoxin. Many times we heard this word carcinogenic and teratogenic. And another interesting one that we published in this paper, you can see normal eye development in the control embryo of the chicken, and left one that we inoculated the in ovo inoculation of the OTA in the embryo, and there is a one eye missing. So these are the teratogenic effect. Those are rendered if you are feeding high level of mycotoxin to the breeding hens. That we published in toxicologic pathology, that is the leading world journal. And right one, you can see the condition of the liver of the laying bird that was consuming the high level of mycotoxin. And these were the histopathological, and the lower one, very interesting, is the bursa. You can see the immunohistostaining in the bursa. This brown color is indicating the presence of the mycotoxins in the bursal follicles. So this is another important study. One of my student conducted experimental study on the male of the cockerels, and he fed the degraded doses of aflatoxin, and you can see changes. A is a normal testis, and then you can see as the level increase, testis volume and weight is decreasing. And the left one is showing A and B. Both these are showing the normal seminiferous tubules with the spermatozoa. But all you can see in the lower one, there are no spermatozoa on the spermatids, and the seminiferous tubules, human is not patent. So it will lead to the infertility in your male. Although you have a specific number of male in the breeding flocks, and if your male is not active, so you need an active male. Normally 7 to 8% male is used, other is as a reserve. But if your male is 7 to 8%, but that is not active, so you are getting eggs but not fertile eggs. And from a breeding flock, you need fertile eggs, not eggs. So, another important aspect of mycotoxin is the immunosuppression, because these are known immunosuppressive agents, because it lead to the decreased body weight to organ weight ratio. And generally, it lead to the decrease in immune organ weight, thymus, bursa, and spleen relative to the body weight. This is called as relative organ weight. And if the bursa size will be decreased, ultimately it will lead to less antibody production, and ultimately it will lead to the immunosuppression. And there will be a lymphocytic depletion in the thymus and bursa, because in thymus and bursa we need rich population of lymphocytes. And if there is a lymphocytic depletion, it will lead to the less production of antibodies and marked damage to the hematopoietic and lymphopoietic tissue. Poor response to the vaccine antigen. Very interesting. You have a good quality vaccine. On the other side, high level of mycotoxin in the feed is coming continuously. Your quality vaccine will not work, because in the presence of the mycotoxin, antigen presenting cell number will decrease, and you need the rich population of APCs to respond to the vaccine antigen. Therefore, it will lead to the less antibody production than immunosuppression. And it will also lead to the anemia that is leading to immunosuppression. This is the immunological parameter one of our students conducted, that he given the sheep RBCs and he collected 7 days, then at 7 days collection and booster dose, and you can see in the A, B, and C, those were fed with the toxin. There was less number of antibody production against the sheep red blood cells. Another important one is that is indicating the macrophage engulfing tendency. If there is a macrophage population will be less if you are a high level of mycotoxin. So we injected the Indian ink. So it is a foreign particle. So macrophages should engulf and clear it. And if there is a poor immune response, this clearance will delay, and ultimately it will lead to the poor response and decrease macrophage engulfing tendency. And another one is very important, as Professor mentioned, about the inflammatory response. So strong inflammatory response is an indication of strong immune system. So we injected the avian tuberculin or PHAP in the one toe of the bird, another was normal. And you can see in the A, there is a measurement of 0.305, and B, C, and D, all having a poor inflammatory response. Those were fed with the different doses of aflatoxin. So aflatoxin will severely affect the inflammatory response of the birds also. These were our paper that we published from these immunological. And this is a recent study. My student, he is also having a talk, Salman. He did in his PhD work in the tunnel assay, and the upper one is histopathological slides, H&E stain, and lower one you can see in the tunnel assay that is indicating with the apoptotic cell with the green fluorescence in the A, B, and C. And similarly, those are seen in the kidneys. These are the apoptotic bodies. So, if we summarize these mold and mycotoxin, what do you need to know? Fungi are throughout in distribution. Most fungi don't produce mycotoxins. Because fungi are even used for the production of enzymes. More than 400 identified mycotoxins and 30 well-characterized mycotoxins are considered harmful to animal and human health, usually ingested in contaminated food. Most are thermostable and cannot be destroyed in normal cooking temperature. Usually no treatment for mycotoxin poisoning. Drying will not completely detoxify the mycotoxins. This was a briefly that our group is working still since 2005. We are working on the mycotoxins, and around 100 students those have their degrees of PhD and MPhil on the mycotoxins. And these were the fundings that we completed. And currently we have two funding, one from HEC and one from Pakistan Science Foundation. Now, few slides regarding the strategies to combat this issue of mycotoxins. There is a decontamination method, physical, chemical, and biological. And if we divide these as pre-harvest, that is a good agriculture practices. That is very important. Timely sowing and timely harvesting of the crops. If you delay the harvesting, ultimately it will lead to the fungal contamination. And favorable storage practices is very important. And cultivation techniques are very important. Nowadays, people use antifungal substances. Some are using the atoxigenic fungi, their inoculation in the crop. Then harvesting is good harvesting practices, and transportation is very important. Then post-harvest level is the sorting of the grains, processing, storage, radiation, and ultimately use of the mycotoxin binders. Among the detoxification, there are two types of products, mycotoxin binders and mycotoxin modifiers. Binders absorb the toxin in the gut, resulting in excretion of the toxin. Modifiers, sometimes they modify the mycotoxin into less toxic products. And among these modifiers, we include bacteria nowadays, yeast, fungi, and enzymes. And these, our group has worked on these substances to identify the locally available substances. Those are having this potential. These are the papers published in the world-renowned journals on the mycotoxins. And we have some work on the silmarine that first time I remember in 2009-10 we imported from the China. Nowadays, this is frequently available in Pakistan. Now, this is you can say this is a weed that is present in our northern areas and even Sargodha. Now, many companies, those are many agriculture farmers, they are just growing the silmarine. And many of us know the silver tablet of the Abbott company. That is actually a silmarine that is very popular in the human for the hepatic patients. So we started working, and now we have developed the product. I will briefly tell about those. This is another interesting thing. One of our students worked on the sea buckthorn. This is actually, it is also named as Siberian pineapple. And sea buckthorn berries, these are rich in our northern areas in the Gilgit-Baltistan. And this is just used for the using the people, those are involved in the preparation of the shampoos for the hair. For the hair protection, those are using this, but this is a very good mycotoxin inactivating agent. And we have developed the product I mentioned about the HEC funding. We have developed the product, and that is now going for the commercialization. With the name of this product, and definitely we will go for the registration of this product. This field trial, experimental trial, everything is completed. This is for the feed mill level. And another product that we developed to tackle the acute mycotoxicosis at the farm level. So this we developed in the PhD thesis of the Salman, and this is a very important, and a comparison is that imported product in Pakistan generally costs $20 to $25 per liter, and it would cost maximum up to $5 per liter. All these substances are available locally in Pakistan. Those are the ingredients of this product. Well, 2 or 3 slides I personally did in my postdoc on the one health aspect of the aflatoxin B1. I mentioned carcinogenic effect. We collected sample from the Faisalabad. We collected sample from the Pinham and other cancer hospitals to check the level of aflatoxin and their relation with the hepatocellular carcinoma. Because due to the shortage of time, I will just briefly tell. We collected the sample of the liver disease without hepatitis B, with HBV, with HCV, and with liver cancer, and with liver cancer and hepatitis B and C. And then we also collected the serum samples. And then we standardized, I did this, some part of this work in the Chulalongkorn University in Bangkok. And we targeted the P53 gene, that is the tumor suppressor gene. And the results in first part, we we amplified the P53 gene, that is a 254 base pair product. The second part was to check the specific mutation. But interestingly, this is a good news that we, although our sample number was less, around 100 sample, but we could not found that mutation in the Pakistani patient that was specifically related to the aflatoxin B1. But those were having, still those were having high level of liver enzymes, because those were having high level of the aflatoxin B1 that was checked through HPLC in their serum of those patients. So our results indicate no aflatoxin B1 specific mutation was detected on the 249 to 50 exon 7 was detected. In current study, only few specific primer were used. Sample collected during this study were only 150. However, it was observed that in all those samples having aflatoxin B1, the hepatic enzymes were at higher limits. And the results of the current study may be considered as a preliminary data. And these were the serum level. I mentioned that aflatoxin B1 was found in the 51 samples out of 150. 34% of these patients were having level of aflatoxin B1 in the detectable limit in these serum samples. So, exposure assessment of mycotoxin is a complicated case. So, this is a very important that we need to have such some planned work in the human health sector on this side. These were our collaborators in the US National Academy of Sciences and Chulalongkorn University, Bangkok. And the credit goes to this person who was my mentor and who developed our lab, Professor Dr. Zargam Khan, who started this research work on mycotoxin in 2005. And he started from scratch. And currently we have all the high level detection facility available in our lab. So I think this slide is salute to this gentleman. And this is my research team that is actually I am presenting on their behalf. This is their wonderful job that they are doing and they are working day and night. And I am thankful to the organizers of this conference for their inviting me and providing this opportunity. And they really, Dr. Deborah Rahman and his team, really, they made a wonderful job. Thank you very much.